So we know from before that cellular respiration is using the products of photosynthesis. So at the beginning, the reaction involves one molecule of glucose, whose formula is C6H12O6, and it's a solid. Now the reaction requires 6 molecules of oxygen gas. You'll notice that I'm writing the word and the formula because you need to be able to recognize both. The products are 6 molecules of carbon dioxide, 6 molecules of water. Now here's something that's new. I'm telling you specifically that 36 ATP are going to be produced. Now this 36 ATP is going to come with a little star. That's what's produced in an animal cell under ideal conditions. The other thing that we get, and we saw this before, is some waste heat. So this is the reaction for cellular respiration. Now before we go any further and talk about the steps, I just want to say a few words about glucose and oxygen, because those are the things that came from photosynthesis. So my question is, do you get to use every single bit of glucose and oxygen you take in for cellular respiration? What do you think? Probably no, since why would I ask you that if the answer wasn't no? I'd like to mention some other things here that glucose can be used for. Glucose can also be stored in a couple of places. So when you consume glucose, it does not mean that cellular respiration is happening immediately. Glucose can be stored in your muscles as glycogen. You might recognize that word from the macromolecules section. Glycogen is a large carbohydrate, so instead of glucose being little individual units, it's a long polysaccharide. It can also be stored as a lipid, so that's fat. Sometimes you consume more glucose than your body requires. If your body is not planning on using the glucose, it will be stored as fat, and that's long-term storage. It's difficult to get stuff to come out of fat once it gets put in. The other thing that could potentially happen is that glucose could be excreted. Now this would be atypical. It is improbable that most of you excrete glucose. Excreted means it could come out in your urine. Does anybody know what might be wrong if glucose is present in your urine? Yes, you knew right away. Something is wrong with your pancreas, probably. People who are diabetic, often the way that you will discover that potentially you have a problem that's in the diabetes category is that there's sugar in your urine. And so this is an unlikely outcome. You would probably have a disorder if that's the case, but it's still possible. So glucose is not all being used for cell respiration immediately. It could be stored and it could be excreted if your body is incapable of handling it. Now in terms of oxygen, we're going to talk at length in future chapters about your respiratory system and about your circulatory system, both of which are involved with oxygen transport and oxygen use. But just so you have a brief idea, oxygen is in your blood and it is attached to hemoglobin. That's something I mentioned already in the functions of protein section. And so for you to be able to utilize oxygen for cell respiration, your blood has to be able to get to the cell that needs it. And so we'll talk in the circulatory section about things like clots and plaque that can build up in your arteries. That would be a reason that oxygen could not be used for cellular respiration. Does anyone have any questions about glucose and oxygen right now that they want to ask? So this is the reaction for cellular respiration. Please note that it's basically backwards from the photosynthesis reaction. Same substances, opposite order. Now this just summarizes visually the fate of glucose, which is what I just mentioned. So this is just a little picture to show you about storage. Does anyone know what the picture on the top that kind of looks like a sideways Darth Vader is? Well, if I look at it, see, this would be like his eye. His other eye could be right there. And there's his hat. Sideways Darth Vader. Anyone know what that is? It's your liver. That's what your liver looks like. So your liver is an additional place besides your muscles that glycogen can be stored. These are pictures of fat cells, and I like to show them to show you that this much of the cell is fat and all the organelles are squished over on the other side. Fat cells are kind of gross. Yeah, fat guy in a cramped elevator. It's a good analogy. The third picture here is a picture of your kidney. That's the organ that we'll talk about when we mention excretion. Your kidney filters things, and it has all these cool little tubes in them. If you're lucky, we will get some fresh kidneys, and we will dissect them. And then the last thing over here is supposed to represent heat and cell respiration, the fact that your body might actually use some of that glucose to produce energy. So, since I went to the trouble of making a little picture diagram of it, I would probably ask you, what are some possible fates of glucose when you consume it, when it enters your body? Plus, I like to show pictures. Eventually, you're going to have to be able to recognize organs from their appearance when we get into the next set of chapters. Oh, sorry. Are you guys actually drawing these little pictures? Fantastic. While I wait for people to draw, did you guys see the little jokes that are on the board? It's a light bulb with "Can I get a watt?" So good. Or the element of surprise. Ah! If it was correct, this was just a joke. But he's right. There we go. Scientifically valid. Now it's like, I feel like I should put an exclamation mark at the end. All right. Yeah, that's what someone said this morning. The sea lion joke was a good one. All right. Anyone have questions about the fate of glucose? A seal is just a neutral sea lion, you know, without the ion. So good. Thanks for that. All right. I'm going to talk about this. In fact, I'm just going to make this diagram go. Whoa, that was weird. What I want you to focus on is the words that are blue, and then I'll talk about that little chart later on. You'll see that the title of this screen is Glucose Oxidation. Oxidation. In photosynthesis, I made glucose by reducing CO2. So it makes sense that when I use glucose, I oxidize it. So remember, I can reduce CO2, but I oxidize glucose. Glucose oxidation is what we sometimes call cell respiration, because that's what's happening. And there are 3 steps. I didn't just draw the box for fun. The box actually represents something. The box here represents your mitochondria. The space around it then represents cytoplasm. So I'm saying that the first step in glucose oxidation, which is called glycolysis, happens in the cytoplasm. Now, what does that mean? Well, it means that you don't have to have a mitochondria to do that step. So this means that any cell can do this step without a mitochondria. You don't need one. And you'll see in a little bit that you also don't need oxygen for this step. So a bacteria that doesn't have a mitochondria can do this step. Any living organism basically can do this step. You don't have to have a specific organelle to accomplish it. Now the other 2 steps happen inside of a mitochondrion. That means that only eukaryotic cells can perform those other 2 steps. A eukaryotic cell, like a plant or an animal cell, has all of the organelles: nucleus, endoplasmic reticulum, ribosomes, and quite specifically mitochondria. That means, for us, I'd like you to remember that both plants and animals can perform this part of cellular respiration. So don't forget, plants do this too. The other thing that I'd like to point out is that what's inside the box requires oxygen. So there are 3 steps to glucose oxidation. The first one happens in the cytoplasm. Any cell can do this. You don't have to have a mitochondria, and you don't need oxygen. The other 2 steps happen inside of mitochondria. That means you need to be a eukaryotic cell, and oxygen is required for those 2 steps. The last thing that I'd like to mention while we're on this slide is that glycolysis, because it doesn't require any specialized organelle, is often thought to be the part that evolved first. We're not talking about evolution specifically in this section. That's for a later unit. But I just wanted to point out that because it's so simple and you don't need a mitochondria, the idea is that it must have evolved first because it doesn't require any sort of specialized cell part to accomplish it. Does anyone have any questions that they would like to ask about the 3 steps, where they happen, or anything like that? Yes. It's another name for the citric acid cycle. So just like the Calvin-Benson cycle could be called that, or the light-independent reaction, the citric acid cycle can be called citric acid or Krebs cycle. Does anyone else have any questions they'd like to ask here? Oops, where did we just go? So we are going to talk about the first step in glucose oxidation. And I'm actually going to maybe split the screen here so that we can look at one thing at a time. So I'd like to look at the description first, then we'll look at the chart that shows what happens, and then we'll discuss where we go from there. So this is the first step in glucose oxidation, it is called glycolysis. So that's the title. And so I'd like to mention the title first. Think about it like it has two parts, glyco and lysis. The lysis part is inferring that I'm cutting something up. If you think about hydrolysis, which we discussed already, that was when water split a molecule apart. Lysis is when we're talking about breaking something apart. The glyco is referring to our glucose, to our sugar. So the idea is that in glycolysis, I'm taking glucose and I'm breaking it apart. So I'd like to go to the description of glycolysis first, so I can give you some details about it. Then we'll go through the little flow chart that shows what each of the steps are. So like I already mentioned, you might not need to rewrite those 2 points. This occurs in the cytoplasm and does not use oxygen. The idea is that we're going to take one glucose, which has 6 carbons, and we're going to split it into 2 molecules of pyruvic acid, which sometimes is just referred to as pyruvate, depending on where exactly we are in the chemical process. The idea is that I went from 6 to 3. Notice how this is basically the opposite of what the Calvin-Benson cycle did. There, I took 2 3-carbon molecules and put them together to make glucose. Now for each of the steps, just like in photosynthesis, you need to know what goes in and what comes out. So what does this step produce? Well, you'll get 2 pyruvic acids, 2 molecules of NADH, and 4 molecules of ATP. I would like to point out very specifically that in cell respiration it is NADH, not NADPH. The one with the P goes with photosynthesis. That's how I would remember it. Now the thing with this step is that even though it produces 4 ATP, you use 2 ATP up. What's really happening is that we have to phosphorylate glucose. It's a little chemical step where we add phosphate to glucose to prevent it from diffusing out of the cell before we use it, as well as making it a little more reactive. So those are the basics about glycolysis. If you remember nothing else, those are the most important things you need to remember. Does anyone have any questions about what happens in glycolysis on a general level or what the products are of it? So then what I'll show you next is a little flow chart that illustrates which molecules are reacting and what is actually happening to get these products produced during glycolysis. Now one thing I'll say, I'm just waiting for people to finish writing here, one thing I'll say is that I would not expect you from scratch to draw every step of the cycle. That would not be my goal, just like it wasn't my goal in photosynthesis. I might use a diagram with empty spaces and ask you to fill them in. That would be a little more realistic. But my goal is that you understand the process that's happening. Does anyone still need the words to be up here? So maybe something else I'll mention while we're here. What's the net result in terms of ATP? Well, since I produced 4, but I used up 2, my net result is that I get 2 ATP. The word net means what do I get when I add everything up? I think I might have mentioned this before. If you have a job, you probably get a paycheck, and on your paycheck it says net pay. That's the pay you get after they take off all the taxes. That's the idea we're discussing here. The word net means overall. If I consider everything that went in and came out, what do I have? So since I had to use up 2 ATP to get this going, my net result is that I get 2 ATP produced. So if we go over here to the flow chart, we're starting at the top with glucose. Glucose is the product of photosynthesis. And the first thing that happens is that I have to invest some energy. So you'll see that 2 ATP come in and 2 ADP leave. That means that we've taken off that third phosphate to make, like I said, glucose more reactive and unable to diffuse out of the cell. Now the next molecule that I get is called fructose diphosphate. I'm not concerned that you know that name. I'm concerned that you recognize how many carbons there are. Glucose has 6, fructose has 6, and then that molecule is going to split into 2. And I'm going to get 2 copies of pGal, which is the same as G3P. That's the thing that is produced by the Calvin-Benson cycle. So the first part of glycolysis is glucose, a 6-carbon molecule. I invest some energy in the form of ATP, and then I end up getting 2 3-carbon molecules. And then each of those 3-carbon molecules undergoes the same process. So perhaps, instead of writing down both sides, you just want to indicate the same thing happens twice. So what happens? Each of those copies of pGal, or G3P, is going to get reduced, or is going to reduce, NAD to NADH and ADP to ATP. So notice here, I start out with NAD, I end up with NADH. I'm reducing. I'm reducing. If I'm reducing NAD, I have to be oxidizing something else. So what is getting oxidized? My carbon molecule. It gets oxidized so that other molecules can be reduced. Now you'll notice that there are numbers. I'm going to get 1 copy of NADH and 2 copies of ATP on each side. So that's how in total I produce 2 NADHs and 4 ATPs. Now my end result on both sides is pyruvic acid, or sometimes we just say pyruvate. And this still has 3 carbons. So notice I haven't taken any of the carbons away. I've reduced other molecules by giving up hydrogens and energy to make phosphate bonds. But I have the same number of carbons that I had to start with. I now have 2 copies of molecules that have 3 carbons each. Does anyone have any questions they'd like to ask about the steps that are happening in glycolysis? So again, you probably want to just write a note to yourself. Same thing happens twice. That way you don't need to write it down twice. The third thing that I'd like to discuss is, where is this pyruvic acid going to go? Because it has 2 choices. And the 2 choices depend on which kind of cell it's happening in. I said on the first slide that you can do this first part without a mitochondria, but what happens next Depends on whether or not you have one. Would anybody like to ask any questions, or does anyone need clarification on any of the steps that are happening here? Okay. So I'm just going to adjust the size of the screen here so that we can see what's on the bottom. So we've got our chart, and then we've got what happens to pyruvic acid. And you have two choices. Choice one happens if no oxygen is present. Choice two happens if oxygen is present. So there's a difference here. If there's no oxygen, pyruvic acid will be converted to lactic acid. You might know about this, perhaps from doing exercise and being aware that while you're exercising, the burning that you feel in your muscles is caused by lactic acid. Just make this a little, oh, that's not better. We'll just scroll down. I guess I'll just have to do a percentage thing and scroll down. Well, that's not better either. There we go, that's better. So if there's no oxygen, pyruvic acid will be converted to lactic acid. This step uses up some NADH. The term for this is anaerobic respiration. That's what we call it in animal cells. The thing that happens is that lactic acid, which is an acid, lowers the pH and prevents muscle contractions. And the very next section that we'll do is about muscles, and we'll talk about this in a lot more detail when we get to that section. If there's not enough oxygen, this process doesn't just have to happen in animal cells. It can also happen in cells like yeast. Yeast cells could produce alcohol instead of lactic acid, and we would call that fermentation. So the process by which, let's say, wine is made is called fermentation. So if there's no oxygen present, pyruvic acid cannot keep going through the steps of cell respiration. Either it'll be converted to lactic acid if we're in an animal cell, or it will be converted to alcohol if we're in a yeast cell. The terms are anaerobic respiration or fermentation, depending on which kind of cell we're actually in. Now, if oxygen is present, pyruvic acid will go to the next step, which is the Krebs cycle or the citric acid cycle. I had both names on the first slide. And this part occurs inside the mitochondria. So evidently, if you don't have a mitochondria, you can't do step number 2. So up until there, does anyone have anything that they would like to ask about what is happening in cellular respiration?